discuss about this Pasteur effect. Myself, Professor Kamthani DC. This Pasteur effect is given by the scientist Louis Pasteur. This is an inhibiting effect of oxygen on the fermentation process, and this anaerobic process suddenly changed to aerobic process. Reduced to cofactors, these are reoxidized by oxygen and more ATP is produced. There is inhibition of the glycolysis by the oxygen. Under anaerobic conditions, glycolysis occurring at higher rate than the aerobic conditions. That means under anaerobic conditions, the conversion of glucose to pyruvate is much more than this aerobic conditions. When yeast cells are exposed to air, the rate of glucose consumption drops. Yeast cells produce more ethanol and muscle cells accumulate lactate. Respiration is much more efficient than fermentation. More ATP is produced and less glucose is consumed aerobically, and this is nearly sevenfold to do the same amount of work in the cell. Pasteur observed that under anaerobic conditions, less growth occurred and large amount of sugar was converted to alcohol. He reasoned that the yeast cells obtaining the required oxygen from the sugar molecule. Yeast grown aerobically exhibit mitochondrial structures, and these mitochondria disappear under anaerobic conditions. When aerobic conditions occur, these mitochondria reappear, and these mitochondria causing the oxidation of citric acid cycle intermediates and also having the role in phosphorylation. Lack of mitochondria, this inhibit the oxidative phosphorylation, and this respiration is changed to fermentation, and this is acting as the powerhouse of the cell, and this is producing more energy through the aerobic respiration. This Pasteur effect is due to the inhibition of this phosphofructokinase enzyme. Due to that, this fructose 1,6 base phosphate further intermediates are not produced, and there is accumulation of this fructose 6 phosphate. Therefore, the inhibition of glycolysis by oxygen, that is aerobic condition, is known as the Pasteur effect. Thank you for your listening.